Hello, everybody. Hello to this special session of Falling Walls, the Berlin Science Week, and JEDI, the Joint European Disruptive Initiative. My name is André Lezoco-Pietri. I'm the director of JEDI, and today I'm with Professor Thomas Hermans. Hello, Thomas, who is our program manager for the JEDI Billion Molecules Against COVID-19 Grand Challenge. Great to have you, Thomas.
Thank you, and thanks for the invitation.
So today the idea is to share with many friends and listeners to of Falling Walls the experiences that we gathered from this first Darpa-type challenge we launched a couple of months ago, focused on the coronavirus. The whole idea of these 45 minutes that we're going to spend together, and please feel free to ask questions in the different chats that you can have, both on LinkedIn, on YouTube, on Twitter, and I'll try to summarize them and then to ask them mostly to Thomas. So maybe a quick introduction about you, Thomas. You're a professor at the University of Strasbourg. You're a chemist, and you are also running your own lab, which is, of course, called the Hermans Lab, which I encourage everybody to follow also on Twitter, not only on your publication in Nature, but also on Twitter at Hermans Lab. And by the way, please follow the news on JEDI on our Twitter account called at EuroJEDI. You're a young scientist from the World Economic Forum. You have had many achievements very early also in your career. So we're very first happy to have you today, and also most importantly happy to have you as a program manager for this very important challenge we launched. So with that, again, for all those who are listening to us, feel free to prepare any questions you will have, and we will make sure we have 10-15 minutes at the end to go into any points that you want to know about this first Darpa-type challenge in Europe. Maybe up to you, Thomas.
Yeah, thanks, André. So, I'm very excited to join today and to be involved in this DARPA-style challenge to tackle or to do something important against the SARS coronavirus 2. But maybe since I got involved at the scientific start of the project, but the project started a bit before, so maybe André can first recap how this challenge came up, and a little bit on the timeline of what were the first steps. Maybe André, you can start with that.
Sure, with pleasure. So actually, so JEDI is launching grand challenges on topics which are too risky or too long term for the private sector. It's a radical new method of funding breakthrough research. We at the difference of DARPA, which is very defense-oriented, we have focused on 5 societal missions: environment, climate change, healthcare. We don't call it digital. The third mission we call it human augmentation, by opposition to human replacement by machines or by digital. The fourth mission is around education. So how can technology and science supercharge the way we learn, the way we do personalized learning? And last but not least is everything around what we call new frontiers, around the oceans and space. And within these societal missions, which are important for our economies, for our societies, for the future of our democracies, we try to identify which are the science and technology bottlenecks that today are not tackled enough, but that could be game changers, and that then later on industry, startup, investors, further research, applied research can push forward. So we really focus on the so-called Pasteur quadrant. So we focus on very clear big problems to solve, but at the same time we remain completely open and not prescriptive on the way to achieve it, and that's why it's so different. We focus a lot on speed also, and we also make sure that we focus on the impact. So we are not here just for advancing a certain field. We're here to achieve to achieve breakthrough results, which is very different than to achieve commercialization. So we are really on the scientific and technology side, and very early on in the process, and we try to disrupt and sometimes really shorten massively. And in this case, in March, so we exist since more than a year now, but we're really operational since early 2020. We are structured as a foundation, and we represent approximately 4,000 scientific and technology leaders in 29 European countries. A big chunk of it is heads of labs, scientists like Thomas. We also have a lot of technology corporates who are represented. We have a lot of universities, academia, obviously, and last but not least, many heads or CTOs of deep tech startups. And all these people help us identifying the right questions to ask, because as you know, it's like a good math or scientific problem or a good philosophy problem. Asking the right question is already the half of the solution. So in March 2020, when the pandemic was declared, we asked ourselves, how can we have an impact? How can we make contribute something different and positive to the global effort that has spun out against this crisis? And very quickly, we identified therapies. I mean, there's a lot of focus right now on vaccines. We identified therapies as another route that is critical to tackle this pandemic. And how could we potentially put together three disciplines which are not enough talking to each other, which is high performance computing, epidemiology, and AI? And how could we not just focus on one miracle drug, not 10, not 100, but could we screen potentially millions, if not billions, of molecules against the virus? So that's how the JEDI Billion Molecule Grant Challenge was created in just a couple of weeks, end of March 2020. So maybe up to me. I mean, let me ask you, Thomas. So once that goal was set, how did we move forward? Tell us a little bit how we structure the challenge, how we define the right question to ask.
Right. So, I mean, the beginning was kind of difficult because my background definitely is not in drug discovery. So there was a steep learning curve on what we could do on a short time scale, and that required us also to look a bit into the different aspects that are important for the early stages of drug discovery. So, as you know, there was a previous SARS-CoV-1 virus, so there were some efforts in the past that have already been ongoing for different targets of the prior virus. But otherwise, there was not that much known about this virus or the different proteins, because what we're looking at here is targeting specific proteins, mainly of the virus itself, and also one of the human host proteins. So one thing that was kind of obvious from the beginning is that there's an enormous space of chemistry that can be explored for drug discovery. So if you think about numbers of compounds that we can synthetically make, it's enormous, right? So in some papers they write there's something like 10 to the power 64 different compounds that can, in theory, be made. So 1 million is 10 to the 6th, 1 billion is 10 to the 9th. So 10 to the 64th is an enormously crazy number. So one of the aspects that we wanted to go after is to encourage the groups that are participating in the challenge to screen at least 1 billion compounds, which is really already a lot for these kind of computational drug discovery. The second point was, like Andre already hinted, is that there is different disciplines along the pipeline that is needed, and they're not really communicating very well with each other. So typically, the first station is the structural biologists that actually purify the protein. They get, for example, an X-ray structure or an NMR structure, so a three-dimensional structure of the protein, and then they post that on hopefully a public repository, and then drug discovery experts, they go and they try to model small molecules that fit in those proteins, and then that's taken on by synthetic organic chemists, which actually make the compounds, and then goes to biophysical chemists or biophysicists that actually test the binding, and like this it goes downstream, and that process typically takes years. And so what we were trying to encourage is to get a good collaboration or a good communication between those different scientists, which we try to do by forming, first of all, a very diverse scientific committee, but also to involve other initiatives that have been, trying to help in the SARS-CoV-2 virus crisis. And the last aspect is that, and I guess we'll get back to this later on in this Q&A, is that the hit rate is terribly bad. So of the compounds that are predicted to bind to targets and to inhibit a certain protein, most of those actually don't work. So it's about 95% that doesn't work. And this is where basically we came in and we copied a model that comes from climate scientists, where there's also there's a lot of uncertainty and noise in the models. But if you put all those models together, you actually get very robust data. So this is, I think, the three main aspects. So many compounds, reaching across disciplines to encourage communication and get the best possible information, and then use consensus approaches to get the highest quality outcomes. So those are, I think, the three main pillars that we were trying to include at the first stages.
Well, sounds very exciting and very new. Obviously, can you tell us a bit more? Who are the partners that we brought in? And you mentioned you hinted a bit about the scientific committee. Could you tell us a bit how it works? How this famous word interdisciplinarity, which is a bit buzzword today in science, how did we really make it happen, and how exciting was that?
Right. So, I mean, as you know, scientists often they operate within their field. So it's difficult to cross the boundaries of different fields. And here, like I said, there is, there's structural biology, there's computational people, there's synthetic scientists. So there's a lot of bridges to cross. But there are people that are able to cross some of these bridges, and we started assembling the scientific committee that really has a very diverse background from virologists, machine learning people, database structure scientists, molecular modeling. There's a very, very diverse team, and everyone knows some of the aspects of the different fields, and in talking to them and brainstorming to people involved in the different aspects, we were able to make those links and to ask people, okay, what's the bottleneck in this step, and what's the bottleneck in the next step? And so we tried to solve these bottlenecks in the best way possible. So that's, I think, the most important thing that we were doing at the early stages. So really identifying the key questions and searching for people that can actually help us answer those questions.
I mean, what really fascinated me, I had the pleasure to participate in most of the scientific committees, is that you are talking about very high-level scientists who are very experienced in their field. Obviously, currently there is a pressure on time. Everybody is very busy trying to find solutions, especially in the crisis. And I was amazed to see that most of them took time every week during the first months of the challenge to be there. So that speaks volume for the novelty and the breakthrough aspect of this research. There are a couple of old friends, if I can call them like that, of Falling Walls. I think Professor Schokop from Max Planck is also part of it, among from Europe, from Asia. Professor Lee from KAIST, I think he also was was a speaker at Falling Walls last year, probably also this year, and also from the Americas. So that was fascinating.
Mm-hmm. Well, one maybe thing that I found very, very amazing is the openness of everyone, all scientists involved worldwide. I mean, it doesn't matter who we contacted, basically everyone wants to help do something in this pandemic. And I think this is, has been noticed on a global scale by all scientists, that the level of cooperation and communication and sharing of information has been absolutely incredible during this pandemic. So I hope we can, use this model also for other big challenges that are still ahead of us. But maybe I can say a couple more words about the scientific partners. So there are other initiatives other
Absolutely.
than, of course, the JEDI Grand Challenge. There is the Coronavirus Structural Task Force that is headed by Andrea Thorn, and they have really gone and into those crystal structures of the virus proteins and actually correct them. So there are little errors that are made when the structures are solved, and those are very important for small molecule drug discovery. And so they have done a huge effort to take those errors out and then put them in the public domain again with, be careful, here's a mistake, it should be this. And so we've been able to also warn our teams to point out, like, these are the best possible crystal structures to start from. And Folding at Home, which is a huge initiative, computational initiative, they have been very active in predicting other binding sites that you cannot directly see in the crystal structures, but that you can elucidate by doing molecular dynamics. So they have done a lot of simulations, and they've provided also these cryptic binding sites to us. It was a little bit maybe late in our challenge, so a lot of teams had already really started on other targets. But I think those new binding sites are also very interesting to be explored. So I think
could you maybe tell us more about these? The principle, maybe for those who are not into biology, the principle of both the targets on the virus, how we identified and how we had made the choice, and how it actually works, and probably also these new developments that you have the static binding sites, but you have also increasingly work, especially headed by Folding at Home that you just mentioned, on these cryptic binding sites, which are actually making use of the dynamics of the molecules.
Right. So there are about 28 unique proteins that the virus expresses once it's inside your body, and some of these proteins can be enzymes. So they are kind of catalysts that work in your body, and they are needed for the virus to reproduce. So some of these, let's say the target that's the main focus of most of the drug discovery efforts at the moment is the main protease. So it's an enzyme that is expressed by the virus itself. Another very well-studied one at the moment is the spike protein. So that's the spikes that are on the outside of the virus that actually make the virus bind to the human cell and then enter the human cell. So what we didn't do is actually we didn't tell the teams which of these proteins they should focus on. So we left it completely up to them to decide what is the best possible target to do drug discovery on. So we left that part also to the team. So it was again a kind of collective intelligence of the teams to select the most promising targets. And as a result, I think we also are looking, so we're looking at 6 targets in total. And so we're looking at a more diverse set of targets than has been done so far in the literature. So I said, the spike protein, the main protease, and the papain-like protease are the ones that have been most studied, because also they are the most conserved from the previous version of SARS-CoV-2. So again, we left it up to the teams to decide what's the best target, and they were free to pursue any target they wanted.
So maybe you can tell a few words about the setup, because you explained very clearly how we came to this clear question to ask, how to move forward, which kind of scientific partners we brought in. It's really a hugely collaborative effort, and in just a couple of days, I mean 2-3 weeks, we were able to build this gigantic scientific committee and partners all over the world. I have to also mention that. Very clear, very rapidly, a few large organizations, especially foundations, supported us, and we were able also to fund this challenge with a very significant prize. So I have to mention here the AXA Research Fund, which has immediately taken up, and kudos to them, but also to many other foundations that supported us. We also had a few pharma companies through their more pro bono work. I mean that supported us, because there's no connection whatsoever between any commercialization and the result of our work. Merck KKR, so the German Merck supports us immediately, and what is amazing, and it's probably due to the pandemic, but also probably due to this very innovative approach. In just a couple of weeks, they committed several €100,000 to this challenge, which is pretty unseen. That means that when you bring something and a radical new approach to a really clearly identified problem, and probably I would add what you just said, is if you leave also the collective intelligence, the freedom to go into any direction that these excellent teams from all over the world consider as good, you can achieve incredible results. So that's probably one of the first big takeaways, is that everything is possible, and so this is also reassuring and positive. So maybe tell us now about the launch, Thomas. How did that work? So we were end of March with this idea. 6 weeks later, we start on May the 4th, of course.
Yeah, exactly. So may the 4th be with you. That was the catchphrase, right? So we of course didn't know what to expect. So we asked the scientific committee, how many teams worldwide, academic or in industry, do you think can actually, do a screening of 1 billion compounds versus a protein target in a very short time, so 1 or 2 months? And numbers were somewhere, I said around 200 teams should be out there somewhere on the world that could be able to participate. But of course, in the beginning, we were a bit afraid that we didn't have the network, the connection yet established. So we were a bit afraid that was not going to work. But through very good dissemination, so of course our scientific committee, they are leading figures in their fields, so they could broadly communicate it across their network. Also, several of these members of the scientific committee are editors on scientific journals that are relevant for this field, and those journals actually also really stepped up to promote the challenge. And also, for example, Nature did a full page ad in their journal to bring awareness to this, and we rapidly saw the number of teams increasing. So it went from a dozen in the first week, and rapidly it was shooting up to the final number, which was 130 teams that were actively participating, and in each team. It ranged from a single researcher up to 35 researchers in the same team. So I think that was very exciting for everyone involved to see that actually the number and the participation was incredibly high for number of teams. So that was I think the first good sign that things were moving in the right direction.
Absolutely. So this number of teams was a big surprise to us, and that's also probably a message to some of people who are listening, asking themselves on other big problems to solve. The key here is obviously that we want to make sure that everybody who can have an outstanding contribution to a given problem, wherever it comes from academia, from the corporate world, from the biotech or the startup ecosystem, if it's more a digital technology, should be able to participate. And so we try to lower the barrier of entry. So we want to make sure that people focus 99.9% of their time on science rather than on filling up, reports or grants demands. We're not saying that that should not be done, but this is how we think a DARPA-type approach, especially in Europe where it does not exist, can have really a contribution. And we were amazed to see that for a lot of scientists, having very high expectation, because Thomas, you will explain to us in a minute what are the targets that we set, which are super high in terms of viral reduction and affinity. But I will let you explain that. So the expectation is super high, and the price is only there for those who can achieve these. So we're not paying for efforts, if I say like that, but really we're looking for breakthroughs. But on the other side, we don't want to transform that kind of research into a more administrative part. So we leave complete freedom, very little. We ask the teams to entirely focus on their science and speed. Obviously, the fact that we were able just in a couple of weeks to do that was the main takeaway here. Is what you just said, Thomas, is it is key when we launch a challenge to make sure that everybody all over the world, in every lab, in every university, in every ecosystem knows about the challenge. And that's where we need partners, we need dissemination, we need scientific journals. And again, thank you to Nature, but to many others journals who really participated and disseminated. We need it even more to make sure that that when there is a challenge like that, that excellence is the only criteria for us. So Thomas, tell us a bit more now the scientific challenges that we launched. What were the criteria? What were the approaches we saw among the teams?
Right. So one requirement we set was that for each protein target, each team should screen at least more than 1 billion compounds. And they should do so using 3 different independent methods. So this is challenging because most research groups are focused on 1 or 2 methods. So when we set 3, it was it's immediately challenging. But the idea was again to have different levels of detail, and this is. This is, of course, already standard done. So it's this kind of funnel of drug discovery. So you have a coarse method first, and then you zoom in more and more. But there were very different approaches, right? So there's the traditional molecular dynamics and docking approaches. So this is really looking how a small molecule fits inside a protein binding site. But there were also about 30% of the teams were using machine learning. So they were basically mining the results of similar proteins from SARS-CoV-1 and then trying to extrapolate this to this billion compounds to select promising compounds that could be tried out later. Then there were, other than that, there was also QSAR, so quantitative structure-activity relationship, quantum mechanical approaches, pharmacophore approaches, and clustering approaches. So it was a very diverse set of methods that were used by the teams. And then, that all together was to find, to reach our main target. So the target is still that we should find a compound which has a very good binding affinity, and with very good, we set a quantitative threshold of 100 nanomolar dissociation constant. So that is actually, this kind of came up in talking to people in the field, and we asked, what would be an absolutely amazing binding affinity, and they said, well, if you can get below 100 nanomolar, then you have a really promising compound that can rapidly be developed. So,
tell us a little bit what that represents, Thomas, maybe compared to other drugs. And it's always difficult to compare because densities and concentrations are different. But tell us what 100 nanomolar represent.
So, for example, remdesivir, which is clinically approved, by this emergency approach, has a binding affinity of, I think it's 1.7 micromolar. So it's, let's say, 1,700 nanomolar. So we are looking for a compound that is, a factor 10 stronger binder than remdesivir. Then how that translates to actual, in vivo activity, that is another point. But it indicates that that is a very high standard. And actually, in a parallel approach, which is also a very interesting initiative, the COVID Moonshot, also Folding at Home is involved in there. They are approaching this 100 nanomolar barrier. It's still not there, but I think they are in the 300 to 300 nanomolar range. So it seems to be possible, and I definitely hope that our teams will blast through it and go to the low nanomolar affinity. But I think we can actually get there.
It's interesting what you say, because often when people talk about moonshots, they imagine things which are 10, 20 years away. Here we really focus on real frontiers that nobody has broken yet, but that are in reach, provided we accelerate, bring collective intelligence, and also push excellence to its best. So we are not there yet, but looking forward to some results. But please continue. We had also a few other criteria.
Right. So So maybe I can tell a little bit about the steps we did So I think for the teams we covered everything. So we had the three in different methods. We had the 100 nanomolar binding affinity, which we will determine experimentally. That's very important. So the simulations, like I said, have a quite high error rate. So 95% of the compounds that are typically predicted, they don't bind or they don't have any activity. So that's one aspect. And then, of course, we encouraged the open sharing and publicly available data. So the methods that the teams use, we really highly encourage them to be openly accessible. We also wanted to encourage teams to have the largest possible libraries that are available. So there are these virtual libraries. So these are libraries of compounds that don't yet exist, but are, probably can be made with high probability. We also worked a lot with the different databases that are available and with Merck to provide the teams with these huge libraries that now exist. So then we had the 17th of July was the deadline for submission. So all the teams submitted their lists. And then there was quite some work to be done by the scientific committee, because, of course, those lists had to be compared. And we used different methods actually to do this comparison. So so we wanted to do different things. One was, of course, we want to respect the individual teams. So we had half of the list, which we are calling now the ultimate list, is composed of the top ranked compounds of each team. So each team has a number of compounds, depending on how many teams are participating on the same target, that translates to a certain number. And the other half we extract from our consensus analysis. And this consensus analysis is very open to discussion on how that could be done, but I basically decided on two methods. One is generative topographic mapping, and the other one is cluster analysis. And we settled on key K-medoids clustering. And so basically, these are methods that map all the compounds in an n-dimensional space, and then they look, okay, where do we have groups or areas in chemical space where there's a lot of agreement between teams. So these are so-called cosmopolitan areas. So we look how many compounds by how many teams that are chemically similar are in the same regions. And then from these regions, we sample the most promising compounds. So that constitutes the other half of the list. And then those lists, these ultimate lists, they were then filtered by different parameters. So we check for solubility, like how likely are they, soluble in aqueous media, which is very important if you are talking about drugs. Also, the molecular weight should not be too high, so that it's difficult to make and very costly. And the synthetic feasibility had to be, and of course the cost has to be taken into account, because some of these compounds can probably be made, but not for a reasonable cost. So all these aspects have been taken into account. And so overall, we went from over 50 billion compounds that have been analyzed by all the teams to 640,000 that were actually submitted to JEDI. Then using this consensus analysis and the top ranking, we narrowed it down to 12,000. And then now we are synthesizing 1,000 real compounds that will hopefully in 2 months be available for analysis. So it's really a big selection, and I hope that let's say our consensus approach will really turn out to be a good approach and really get those hit rates much above 5%.
Thomas, there is a question in one of the was asking, maybe it's a good moment to bring that up here. That somebody's asking how much you mentioned that this is similar to what has been done in climate models. Could you tell us and elaborate a little bit more on that, and why is it innovative? Has it indeed never been used outside of climate?
So, I mean, I think so. In climate models, of course, there's the International Panel on Climate Change, the IPCC, that looks at these models and compares them and does predictions on robustness and reliability of each model. And overall, I mean, scientists now are I think 97% convinced that climate change is due to human action, and not just because the sun is doing something weird or any other aspects. I think what makes it more difficult in drug discovery is that a lot of drug discovery is done by pharma, and they're absolutely not open, of course. So they're not going to share, all their possible compounds that they are considering because of IP reasons. So I think that this model has not really been viable for drug discovery, just for I think mainly for IP reasons. And here it's different because people are very motivated to do something about the pandemic, and also willing to freely share their information with each other. So I think this was a kind of excellent opportunity to try this method, but under normal circumstances, I don't think it would work. That's the big difference.
So this means that I mean the topic of IP, we didn't mention that yet. I mean you just it's indeed critical, because basically the products we have is on one side. And it's another question I have for you from the chat is from the listeners to this discussion is how much did we? The question that I just see here is how much did we use this concept of collective intelligence? You say you have 130 teams, but how did we make sure? Maybe you can elaborate a bit on the few symposiums we had, and we tried to have teams talk to each other. But the other question is, how much do we make sure that on one side we have open science, which clearly disseminates information much faster, and on the other side We are talking about the preclinical drug discovery. Somebody that needs to be incentivized to put still the money in order to have the clinical test. Otherwise, we create great results, but we end up with drugs which are not tested and which may never hit those who really need it. So I would love to hear you on both aspects.
Right. So for the teams, the way we tried to get communication going is we had a shared chat. So we had a Discord channel where all the teams could go on and offer their expertise or ask for missing expertise. So several teams actually merged using this platform because they found each other and they said, okay, you have these two methods, we have this method. So they joined forces. And in the webinars, we had in total, I think, somewhere around 10 teams over the different webinars that briefly presented their methods. Of course, they were a bit reluctant in the beginning to share their final compounds because at that stage, it was still, the competition was still open. But after we closed, that's what we're trying to get to now that the deadline is finished and we have finished our analysis, that we now make this discussion much more open, to see these are the final results. As soon as we have the experimental values, we can really have an open discussion with all the teams to see what we can learn from this and how we can proceed from this point onwards. So I think that's quite interesting. For the second point on IP, it's a very difficult point. So if when we were talking to the more traditional pharma, it was absolutely out of the question to put these kind of results in the public domain, because it would possibly make the further development more difficult. But there are other opinions, and on one of our webinars, we had Alad Edwards from the Structural Genomic Consortium, that is a big supporter of open science, and especially at this very early stages. So we are really doing hit discovery. So we're trying to find completely new compounds that can have an activity on the different targets. And there's still a quite long road of development of lead optimization and other aspects that have to be considered. So there should be still enough IP left for traditional pharma to use. And then, of course, we should rethink maybe the entire model of who needs to pay clinical trials for drugs. Maybe if that can also shift more to a publicly funded model, then there maybe the openness and the sharing would actually accelerate things much more. But there is, of course, a huge bill to be paid by someone at some point along the whole trajectory, and that's still a difficult issue at the moment, I think.
Fascinating. I mean, I want to stress this part on IP to demonstrate how much pragmatic we need to be on that one. And this is why we really believe in the mission of JEDI, is that we need to be extremely pragmatic and science-oriented in these, especially the pandemic has shown to us that every day, every week counts, and that speed as well as thinking about the next big thing is so important, because if we are looking at the battles of today or of yesterday, we always run after. And this image, I think speaks to everybody when we see what's going on in the current situation. On the collaboration of teams, I just can't stress enough what Professor Herman just mentioned, is we all know that maybe traditional research is often done in consortia, and when you look at traditional grant models, they always look at a certain number of participants. Sometimes it makes sense, sometimes it does not make sense. What we just say is that sometimes a team just needs to run and will have a breakthrough alone. Sometimes it makes sense to combine. But if you predetermine that, the risk is that you put people into too narrow thinking. What we try is to have both competition, which makes people go fast and and get beyond their own capabilities, the best of themselves, but also cooperation. And you said it yourself in this symposium. Initially, people were a little bit close to the chest and they don't want to disclose, but then they realized that sometimes by combining forces, they had much better chances to succeed. So we'll try to do the same in the upcoming grand challenges we have. I have another question, but maybe Thomas, tell us. You mentioned now narrowing. Tell us about the next steps. Where are we now? What can we expect from this grand challenge?
Right. So at the moment, as I said, we selected 1,000 compounds that will be synthesized. So we have a partner organization that is going to do all the synthetic work, which is really a huge amount of work. If you know that a typical chemist can make maybe 2 to 5 compounds in 1 week, so 1,000 compounds is a lot of man hours of work. So we're very happy that our partners are committed to make that happen. And then those
this point needs to be stressed. I mean, synthesizing 1,000 compounds. I don't know how many people are doing that currently on the globe for the pandemic, but probably not so many. I mean, this is a huge endeavor, and hopefully it will bear fruit.
Yeah, exactly. So I mean, you need either thousands of chemists or a lot of chemists that work a very long time to achieve that. It's really a very daunting task. And hopefully in the future there will be more automated ways to do this synthesis. But for the moment, a big part of it remains very manual labor. And so once we have those compounds, we will check the binding constants of those compounds versus the different targets, and also for the proteases. So for the main protease, the papain-like protease and TMPRSS2, we will do a cleavage assay. So there we look if the enzyme can actually cut a probe with 2 fluorescent markers, so we can actually see if it's enzymatically active. And that screening is a bit faster than the binding studies. So we hope to do very rapid analysis of these results again with some of the partners of the JEDI Grand Challenge. So hopefully by end of January, we will have the binding constants of all those compounds, and then the winner of stage 1 will be really determined by the experimental validation of the different compounds that have been predicted by the teams. And then once we have all that data, we hope that we can enter stage 2, where we will look at really the viral suppression in live cell models and hopefully animal models. So really put those compounds on cells that have been infected with the virus and see, can we get a 2-log reduction? So 2-log reduction means 99% reduction of the infectious yield in a cell or hopefully an animal model. And that is the next big step that needs to be taken to get those things further, eventually to the clinic in the longer time in run.
Can you tell us how much this 2-log is a real breakthrough compared to existing drugs, and what they achieve in terms of viral suppression?
Well, you can of course get a big suppression if you put a huge amount of your drug, but then at some point any compound will become toxic, so cytotoxic. So the trick is really to get viral suppression selectively, so using a small concentration of drug, which really selective is on the virus, so you get good suppression without getting a lot of side effects. And at the moment, we still don't have a good compound that actually directly targets the virus. So even remdesivir, it binds and it improves a little bit the recovery time of patients coming out of the hospital, but it doesn't actually help for the mortality rate. So that is really still a problem. So we need selective therapeutics for this virus and also future viruses to really alleviate the pressure on the hospitals. So of course the vaccine is very important, and hopefully will save us in a year or so. But still, you will need hopefully a low cost and accessible therapeutic to work on the virus.
We are coming to an end to this discussion, so thank you so much for these insights. I have a last question that come up in one of the chats, which is about, well, it's a bit related with what you just mentioned about the vaccines and therapies. It's now the number 5 coronavirus that has an impact globally. This one obviously much more than all the others before us. First, we need to underline there has never been a vaccine successfully developed against one of them. There will be a winner of our grand challenge, at least we hope, if they pass the threshold. If we don't pass the threshold, we won't. But there is also a humongous amount of data, as one of the listeners to us. I mean, has this any value? Can this be used to train, especially machine learning, the application of AI into drug discovery? How do you see that as a building block that also this JEDI challenge can help?
Yeah, definitely. That's a very good point. So definitely all the data that we are going to generate and will put in the public domain once we have all the experiments can then be used again by teams to improve their binding studies, and basically it's a feedback loop to the community to develop even better compounds and repeat the process and actually accelerate it. So for machine learning, they're very good at extrapolating information, but if you don't have good initial information, then it's very noisy to do these approaches. So definitely what we will contribute also to the whole field and for future coronaviruses is to have a big data set of experimentally validated compounds that have an activity. So that is a very valuable resource for anyone in the world to continue on these therapeutic developments.
At one of our symposium, Sir Peter Ratcliffe, with the Nobel Prize 2019 in medicine, who was part of the JEDI scientific committee, we discussed a lot about what can we learn out of this effort that we've been doing, and many other efforts you mentioned for pandemic preparedness, because we see how costly it is to be in this, costly humanly and economically also to be in this crisis, and how can we prepare? How can we prepare against future coronaviruses? We don't want to spread fear here, but it's probably not a question of if, it's a question of when, especially given the proximity now with and the biodiversity issues. We don't know exactly if there's a relationship, but there seems to be. To tell us how you would ideally see this community that has been built around this challenge, these 130 teams, all these scientists that are working so positively. What would be your dream what could we do out of this community?
Well, I think so. I mean, we see even that now that we are going faster, maybe through the JEDI challenge, the response time is still too long. Let's say we cannot have a pandemic and have a therapeutic drug on the market in 6 months. So even though we want to accelerate as much as possible, there's definitely a limit to what can be done. But of course, I mean, there are scientists that are studying all the possible viruses that have a propensity to jump to the human population. So we could map those viruses and do similar approaches versus viruses that have not yet jumped. And then when one virus, a zoonotic virus, jumps to a human population, and then we can hope that it will be similar to something that we already are studying in advance. So it's I think reacting fast is important, but also being prepared and looking for what are the next possible viruses, and actually doing a full drug discovery against those viruses. So we have a library of information ready for the next one that comes, so we can actually react on a much shorter timescale. But just like for IP, it's the question is who's going to fund that type of science, because there is there's no market yet. No one wants to yet pay for it, but it's very important, more important to be well prepared, and it's much cheaper to do that in advance than after the fact, as we are seeing now.
Professor Hermans, I think this call has been well heard by those who are listening to us. I must say that several of the partners that we brought in for this challenge were partners that we met during the last Berlin Science Week and Falling Walls. So it's always a great moment for good-willing people who want to advance science and technology to come together. So don't hesitate to reach out to us. Thomas, thank you so much. It was super inspiring. And if any of you wants to get in touch with the Joint European Disruptive Initiative, just ping us. It's JEDI@jedi.group. Follow our Twitter account. Follow Thomas' work, because he's not just doing the Grand Challenge. He's also doing amazing work on microfluidics, but that will be another opportunity to talk about that. On the program of two last things. Tomorrow we will have a session on probably our next Grand Challenge, which will be on green hydrogen. We will have an expert discussion on the frontiers. It's going to be at 1 p.m. European time on Monday, November the 9th. And also stay tuned, because everything that Professor Hermans just mentioned on the preparedness, we will probably move forward on the pandemic preparedness initiative and try to join forces with many others who are working on that. If we can contribute the library that we built up, this amazing amount of data, we'll avoid what apparently happened this time, is that we start the work from day one. And I remember some of the teams told us what was missing to train our model is the data that we could have from SARS-CoV-1, from MERS, and so this is what to make sure we progress each time. So, Professor Hermans, thank you.
Exactly.
Thank you.
Yeah, thanks a lot, Andre.
And see you all very soon. Thank you.
Yeah, thanks everyone.